Welcome to my presentation about spatial cellular networks from omics data. My name is Michael Altenbuchinger. I'm from the Department of Medical Bioinformatics from the University Medical Center in Göttingen in Germany. It's really my great pleasure to present here at the 10th Annual Cancer Research and Immuno-Oncology Virtual Week. And I'd like to present you about graphical models, and these models we use to estimate molecular networks from high-dimensional omics data. So definitely a topic of huge importance in the context of cancer. And I'd first like to make you familiar with the concept, how it works, and why we think that it's at least a great concept which has multiple advantages compared to standard correlation-based approaches. And I particularly want to show you how this framework can be extended to the multi-omics content. And specifically, I want to show you an application where we integrated transcriptomic data with promoter methylation in breast cancer. And basically building up on these models, I finally would like to show you how they can be adapted to also analyze spatial omics data. So many people are currently talking about spatial transcriptomics, and indeed there are not yet many approaches available to basically perform a holistic integration, so including as many information as possible into the models. And that's basically a way we propose, or many people currently are pursuing such directions. So in mathematics, that's really a field where huge of efforts were done in the last few years. And basically, we are very interested in now using that knowledge to gain knowledge from multi-omics data or spatial transcriptomics data, for instance. Let me first make you more familiar with the concept. And for that purpose, I did here some introductory simulation where I compare ordinary Pearson's correlation with partial correlation. And partial correlation, that is a graphical model. Specifically, it corresponds to a Gaussian graphical model. And what I did here is that I simulated two genes which are regulated by a single, I called it here regulator, which might be some transcription factor or whatever. And I simulated the scenario shown here in the upper left corner. So this regulator determines the expression of two genes A and B. And here on the diagonal, you have histograms over the values which I simulated here, and corresponding scatter plots are shown in the upper right corner. And now a correlation-based analysis quantifies basically the scatter plot into a number. And therefore, I put here a question mark in the upper right corner that you just ask yourself, how would you now in such an easy simulation expect the scatter plot between gene A and gene B? And let me resolve it. So if we go on one slide, that also nicely scatters on a line. But that's pretty much in contrast to the simulation we performed, because we basically A and B are not directly dependent. However, considering just A and B in isolation, such a scatter plot, they appear to be some kind of correlation. And that's simply because of the individual relationship of gene A to the regulator and gene B to the regulator. Also, they do not have a relationship in between. And that's quantified, for instance, by Pearson correlation. And I added the corresponding numbers here. So gene A and regulator correlate highly with 95%. and similarly regulator gene B, and the number which I indicated here in red. That's the correlation between gene A and B, and that's still highly significant with 91%. That means, if I go on one slide and depict the corresponding graph, that appears to us as there would be a relationship between A and B. And that's basically what partial correlation can address. So I simply calculate here now partial correlations. And we see gene A regulator still highly correlated with 0.68. The same for regulator gene B, 0.68, and both highly significant. But the red number, the rho, now decreases to something near 0. So also they lie almost on a line. Basically, the partial correlation diminishes. That means partial correlation can resolve the basic structure of what we simulated. Also, I have to emphasize that there are no directions. So these partial correlations does not tell us if A regulates the regulator or if the regulator regulates A. So that's something we do not have here. But what it does is basically that it considers variables simultaneously and allows to disentangle direct from indirect relationships. And of course, in cancer research, that plays a huge role, because in cancer, many things might differ from normal tissue, for instance. And basically, it's of huge importance to get rid of a lot of false positive associations. And that's what these kind of models aim to do. Let me briefly summarize. So these vanishing partial correlations, these correspond to conditional independence statements. So that just conditional independence basically phrases what I just showed you. So this distinguishing direct from indirect relationships. And I also want to express it formally, at least once, or even that's not perfectly formally, it's more an intuitive statement in the corresponding formalism. And that basically reads, if A and B are conditionally independent given C, so in our previous example, C was the regulator, then A does not provide any information for B which is not already contained in C. And that's basically written here on the right side as a formula, and that corresponds to vanishing partial correlations. And basically these graphical models aim to estimate conditional independent statements. And computationally, that means that we need to consider all variables at once. So we cannot consider two variables, for instance, two gene expression levels in isolation, but we need to consider as much as possible, which can help to basically reveal if is it now a direct relationship or an indirect relationship. And of course, there might be in nowadays omics data, there might be still a lot of variables hidden. And of course, that still induces many false positives, let's say it like that. But as I tried to show you, basically we reduce at least the number of false positive associations. And one thing is worth mentioning. So we are usually dealing with high-dimensional omics data, so really many molecular variables. And as I mentioned, we need to estimate really a lot of parameters. And what I show you here is a plot where I have on the y-axis the difference between an estimate of the partial correlation matrix and the ground truth. And the standard partial correlation estimates that's already almost 100 years old and known how to calculate it. That's shown here in red. And on the x-axis, we have sample size. And what we basically see is we decrease sample size and it gets more and more difficult to correctly estimate these partial correlations. And therefore, this red line basically increases. And it increases until it reaches a sample size of n equals 100, which corresponds to our simulation with 100 variables. That means if I have even less measurements or the same order of magnitude measurements as I have variables, I cannot estimate it, or if I have slightly more measurements, the error gets pretty large. So I usually need much more measurements. Measurements mean samples than variables. And that's of course something which is widely feasible. So if we are dealing with bulk tumor tissue, for instance, really large data sets have something like 1,000 patients, and I'd like to model, let's say, 10,000 or even more RNAs or different omics layers. So the red one here definitely causes quite some issues. Where now a lot of research happened in last years is how can we still infer reasonable partial correlations, and that's what people usually do is a so-called regularized estimate. That's basically you induce a systematic bias on your estimate, which basically tells the algorithm, I do not expect too many meaningful associations. So it basically biases all towards simplicity. And such methods, so the blue line here corresponds to the so-called covariant shrinkage approach. So one specific kind of regularization for graphical models, and at least the error does not inflate it uncontrolled. And basically we can also estimate partial correlations for even lower sample sizes. In the lower left corner, I also mention a reference where you can have a look into if you want to find out more about this. And there's also some code where you can generate similar plots. Well, now, of course, there are a lot of new directions coming up, and multi-omics is one of the most promising directions experimentally, in my opinion. And that also generates a lot of computational issues. And basically, we and many other groups try to solve these issues. And I just want to give you an idea what these issues are. So for instance, in multi-omics, we might deal with very different data modalities. So we might have continuous data like gene expression, or gene expression could be also incorporated as count data. So if I really count transcripts, or I have something like categorical data, so for instance, mutations, or could be even something ordinal. And one direction one could go here is mixed graphical modeling, where I mention here also 3 citations. And what we specifically investigated is multi-omics for continuous variables. So there might be still a lot of issues like very omics layer specific noises, or very different amount of edges between omics layers, within omics layers. So edges means associations. Sorry for not mentioning this. And what we suggested is a method we called DRAGON, which stands for Determining Regulatory Associations Using Graphical Models on Multi-Omic Networks, where you can also have a look into a preprint given here under this archive number. The method computationally looks like this. I will not explain too much details about how DRAGON works, but at least I would like to mention 2 publications which were really important for developing DRAGON. That's here the Schäfer and Strimmer paper from 2005, which introduced covariance shrinkage as a method to estimate partial correlations for high-dimensional omics data. And then the second one is here by Bernal et al., where corresponding null distributions were stated. So null distributions, it's something we need to estimate p-values. And those two methods we combine to a multi-omic setting. And that's what's basically what DRAGON is, and in formulas written, it's here shown in the upper right corner and comprehensively shown in the preprint, which I just mentioned before. Well, when we set up such a new method, we also need to comprehensively benchmark it. And the first step we do is we do some simulation studies. And basically, we did 4 different studies. And I'd like to start here with study A. And what you see here on these figures on the y-axis is basically the performance in recovering the correct associations in terms of ROC AUC. And on the x-axis, it's corresponding sample size. And simulation scenario E was constructed in a very benign scenario where we basically have 2 omics layers, and those are basically equal with respect to their characteristics. And in that case, we observe here such a blue line, and what's hidden here is a red line which lies below. That's in principle this covariance shrinkage-based estimate of partial correlations. And as we see, both perform identically. So if I have a very controlled setting where I do not care about multi-omics issues and I just deal with both as they would belong to the same omics layers, DRAGON and GGM, that's basically the same. But it's getting more interesting if we now introduce systematic differences between omics layers. As I said, there might be huge differences, for instance, with respect to feature size, so number of variables, and also edge densities. And that's what we systematically explored in simulation B2D. And there we see that indeed DRAGON improves quite substantially, as indicated by the blue line, which usually lies substantially above the red line. We also benchmarked it against other available approaches to estimate Gaussian graphical models with significance levels. And basically all these methods ranked edges according to false discovery rate, and that generated these plots. The first row is what I just showed to you, comparison to basically we could call it one layer DRAGON here in red, GGM, and then second to fifth row are the competing methods. And there basically we see a very similar pattern for each of the comparisons that DRAGON indeed, through explicitly taking into account this multi-omics aspect, indeed is able to improve the network inference substantially. Well, I'd like to present you now an example analysis. And there we considered breast cancer specimens from TCGA. And first, I'd like to give you just some sanity checks, which also emphasize how a partial correlation analysis can indeed improve compared to a Pearson correlation-based analysis. And what we did here is we downloaded these breast cancer specimens from TCGA and restricted the data set to gene expression levels of transcription factors and corresponding promoter methylation levels that made roughly about 3,000 variables, which we included into the model. And now performing an correlation-based analysis, we obtain a huge amount of significant association between methylation levels and gene expression levels. In total, we obtained more than 800,000 significant associations with a false discovery rate below 0.5, basically indicating there are a lot of dependencies and associations, but as I said, those might be a lot of indirect ones also. And also importantly, just a minor proportion were edges between the expression of a gene and its corresponding own promoter methylation. And that's what we basically supposed to be the dominant effect. So what's basically happening is you usually have a methylation of the promoter, and that represses gene expression. And that's also why most of these edges were negative. But as I said, a rather low proportion of that, basically what we would consider the most natural way how methylation affects gene expression. Okay, let's see what DRAGON is doing. First, we observe that DRAGON substantially reduces the number of significant methylation gene expression edges. And in fact, there were just 769 left with an FDR below 0.05. And what we observe here is that a high proportion, 43%, in number 333, were associations between the expression of a gene and the methylation of its promoter. And in also the a high proportion of 92%, so the predominant proportion, were negative partial correlations. So that really looks like we have revealed this process of repressing gene expression via promoter methylation. So far, that's a sanity check, let's call it like that. So that's nothing particularly exciting. It just shows you a drawback of correlation-based analysis. But we are, of course, more interested in learning something about cancer. And indeed, I don't want to hide some nice discoveries. So we screened gene expression variables according to the number of edges to methylation sites. So basically, we screened for gene expression methylation hubs. So genes which basically associate with as many methylation sites as possible. And that's yielded a zinc finger protein as top hit. It's CFP57. And first, we observe that the strongest edge in this neighborhood of CFP57 is basically its own methylation site. And it has a really low FDR below 10 to the power minus 100. So really a clear association, and it's negative. So that's very likely that it's really this effect I just mentioned of gene expression repression via promoter methylation. Then it's followed here, for instance, by SCORE2 or another zinc finger protein, CNF121. And now we have this gene expression variable CFP57, and it affects a lot of methylation sites, and nicely CFP57 is also known to contribute in methylation memory of parental origin, and it controls DNA methylation during the earliest multicellular developmental stages at multiple imprinting control regions, as written here. And nicely also in the context of cancer, because we see basically this effect directly in breast cancer specimens. It's also nicely that CFP was shown to suppress proliferation of breast cancer cells here in that publication shown at the bottom. The next one in this list of hits was again a zinc finger protein. It also connected to 11 different methylation sites. Again, it's controlled by its own promoter methylation. And nicely, CNF334 was also identified as a tumor suppressor in triple-negative breast cancer. Recently, as shown in this publication in the third, under the third point, And now Dragon suggests a mechanism with this very dominant edge to its own methylation site. And that's in fact something, a pattern which is already observed in other cancers like hepatocellular carcinoma. Finally, we ended up with this analysis by a data integration or a community detection approach, and that identified a joint biomarker which combines methylation with gene expression. And basically, we observed that we can differentiate basal versus non-basal subtypes of breast cancer with an AUC ROC of 0.85. So that's in fact, considering that it's just one gene expression level and its corresponding promoter methylation, that's indeed a pretty good number. Of course, if you incorporate many more variables, you might get substantially better, but it's remarkable that it's just these two variables. So basically a multi-omics synergy, which we here discovered. Well, the last few minutes, I'd like to show you how these concepts, so basically the concept of graphical model, can be adapted to spatially resolved omics data. And first, I'd like to just state some basic things all of us know. So cells send chemical and mechanical signals. That's pretty clear. Cells receive these signals, and of course, if they receive these signals, there will be also some downstream effects. And that basically leads finally to that the cell adapts its molecular phenotype. In other words, so if I put, for instance, here an additional cell, so I added this dark cell, this cell might look different than in isolation. And that's basically simply because cells talk to each other. And in other words, the molecular phenotype of a cell depends on its environment. And even more, that means the combination of cells' molecular phenotype together with the spatial context allows us to better understand how cells communicate with each other and how they organize in space. And that's what we try to estimate. And basically our strategy was to extend that language of partial correlation, graphical models to the spatial context, and basically to the spatial omics context. And let me first recapitulate what I showed pretty much at the beginning of my presentation. I introduced some language of conditional independence, and there I mentioned that molecular variables A and B are conditional independent given C, then A does not provide any information for B which is not already contained in C. And I just repeat it here because we now use the same language to basically formulate what we call spatially conditional independence. And so in other words, I have molecular variables now in different cells. So for instance, a molecular variable A in cell A. So variable capital A in cell small a, which is here shown as A, capital A with superscript A. And what it reads here is molecular variables A in cell A and variable B in cell B are spatially conditional independent given the rest. Then A does not provide any information for B, which is not really already contained in rest, of course, in cell A and B, respectively. And the corresponding way of expressing this in formula is shown here in the middle. Importantly, we need to specify what rest means. So now really rest refers to all variables of cell A except capital A and all variables of cell B except capital B. And of course, all variables of all remaining cells. So basically, that was our strategy. That's what we now wanted to encode into a model to infer cellular communication. So that's it's not so straightforward to get used to this terminology, and I really recommend you to look into a preprint which we just put on a preprint server a few days ago, which I show you in a moment. So, however, also we can state such a terminal. Also, we can name something like spatial conditional independence. That does not yet mean that we have a model we can easily calculate. And in fact, that's rather tough because spatial interactions are pretty diverse. And so, for instance, they can be a short distance or something like cell-cell contact, or they can be long distance, so exchanging signaling molecules, something like that. And that's now the preprint which I mentioned. We called it SpaceNet for spatial cellular networks from omics data. And the idea is to basically encode the spatial conditional independence relation in an upscaled version of this Gaussian graphical model. So computationally, that's inflates the problem tremendously. So it's p times n, a multivariate normal with p times n variables, but basically we stack the individual cells profile together and try to model it within one single big multivariate normal. And then what remains is basically that we need an doable and reasonable way to encode all these spatial associations. And what we did here is something like making a very general series expansion, which allows us to model very different interactions like cell-cell contact, and also something like more long distance via some signaling molecules. And basically, to show you that this works out, I just, again, in a simulation study, show you here these red lines in figures A to D. These are potential, so basically how one molecular variable in one cell affects another molecular variable in another cell. And that's basically through observing this again and again. So basically, if I have such an image slide or spatial transcriptomics data where I really have individual cells with their location, then I basically observe cells many, many times with different distances, different locations, and that allows me to estimate that with these relationships. And so, for instance, here from left to the right, something like intermediate range is figure A, long range figure B, short range figure C, and figure D is something strange, which we also tried. So something like there's no molecular interactions if cells are very nearby. So there's a potential is near zero. And then molecular variables affect each other across space for reasonably large distances. And then again, the interaction disappears for large distances. And in principle, even something like that can be reconstructed. And the different lines that I show to you here are different orders in our series expansion. And in principle, through increasing orders, we can model more and more complex relationships. I'd like to end up with an illustration how that basically works. And now we went a bit away from cancer and considered data from the mouse visual cortex, simply because appropriate data, so that means a position in space and corresponding to that position in three-dimensional space, also in RNA expression, that's still something which is pretty rare. And in this data set, there were 28 RNAs measured in 30,000 cells, together with their respective location in a thick tissue section. And that's something that's where SpaceNet is built for. And applying SpaceNet yielded these spatial associations between gene expression variables here shown in this figure on the right side. And I just want to mention our top hit. And the highest spatial association was observed between myelin basic protein gene, MBP, and FMS-related receptor tyrosine kinase 1, FLT1. And indeed, that fits nicely into the literature. So myelin, so that's basically built to a high proportion by myelin basic protein. This myelin surrounds nerve cell axons to insulate them and to increase the conduction of electric impulses. And now FLT1, on the other hand, encodes a member of the vascular endothelial growth factor receptor, VEGFR family, and these mediate diverse cellular communication signals controlling developmental processes. And now SpaceNet resolves that FLT levels are negative, so I consider a specific cell, and then FLT1 levels are negatively associated with MBP levels in neighboring cells. So that's basically a kind of statement SpaceNet provides us with. And indeed, that also fits nicely into the literature. So here in a part of the adult mouse, it was shown that oligodendrocyte precursor cells contribute to focal remyelination, and VEGF signaling might be required for the proliferation. So it is known that these OPC. So one thing, of course, SpaceNet just generates, let's say it like that, hypothesis or indicates mechanisms, and at least it's nice to observe this very strong negative association, because it could be some mechanism like there are, for instance, axons which lack MBP, and then basically OPCs, which are the remyelinating cells, group around these axons basically to remyelinate them. That could be one mechanism. Of course, to further elucidate it, that might require substantially more work. And that's a very methodological work. So we are pretty much at the beginning. We now have a model which incorporates all these aspects. And now we hope that we are looking forward to more and more omics data coming up to try to generate really knowledge. And I also want to emphasize, because that's pretty much in the spirit of how my collaborators and I are working, is that we basically try to get a more holistic view on our data. So our data have many, a huge amount of information. That information needs to be integrated as complete as possible. And these network approaches are some kind of way of doing this. Let me briefly summarize. So I presented you first some basics about graphical models. Many groups in the world have contributed. And we have here two preprints out. So Dragon, SpaceNet, which I presented both to you. So also, I'd kindly like to thank all my collaborators. So it was really necessary to combine forces to develop these rather complex models. And of course, I'd like to acknowledge funding from these two funding agencies, BMBF and DFG in Germany. And I finally want to thank you for your attention, and I'm very happy to answer questions.